tighten and arithmetic, taught to the tune of a hickory stick. You were my queen in calico, I was your bashful barefoot beau, and you wrote on my slate, I love you, Joe, when we were a couple of kids.
1.
Lab number 8, the guard cell. Hello everyone. The purpose of lab number 8 is to learn how to prepare and identify both a guard cell and stomate, and to learn about their amazing function. A stomate is a pore. Stomates are found in leaves, stems, and other organs. It controls the rate of gas exchange. The pore is bordered by a pair of specialized guard cells that are responsible for regulating the site of the stomatal opening. Air enters the plant through the stomata pore and contains carbon dioxide, which is used in photosynthesis. Photosynthetic guard cells have chlorophyll. The oxygen from photosynthesis is used for respiration and is produced as a byproduct of photosynthesis. Also, water vapor exits the leaf through the stomata or pore as transpiration. Materials. This is a balsam flower. You need to take a leaf off of the balsam flower, which you may find in a garden store, or you might have it as a plant in your yard. If you can't find a balsam leaf, then you can try a basil leaf, and that comes from the basil plant, and that you can get from a grocery store, the plant itself. The underside of a leaf, the thin white membrane found between the epidermal layer, a clean cover slip and slide. You need forceps, a clean slide, and a cover slip to put over the thin white membrane. So you don't need that many materials. This is all. You don't have to stain anything. You can use saffron, but I'm going to do this just as a wet mount without any dye. Using your forceps, you can peel away the epidermis of the backside, lower backside of the leaf. See the white membrane there? That's what you're going to be looking at under the microscope. Put it on a slide. And then just put a cover slip over it. It's time to observe the guard cell under the microscope. I start focusing, look at 4x, scanning, moving up and down, looking for the white membrane. Here is the guard cell. As you can see, it's a close-up. The arrow is pointing to the actual cell that is guarding, and you'll see like a slit in the middle. That's the pores. As you can see, the guard cells are under the microscope. And I'm going to move it up to 10x. This particular guard cell came from the basil plant, and that's the plant that we eat a lot, and it's found in grocery stores. You could substitute this instead of the balsam. If you can't find the balsam, you can use basil as well. In this instant, you can see that the stomate is wide open, and the water goes through those two pores there that you can't see, but you can see the pore, but you can't see the actual two that are there. On each side is the guard cell guarding the opening or the pore or the stomate. For more practice, we're going to now take out our biology notebook. This is lab number 8, the guard cell. We're going to observe the guard cells and record them in our lab notebook. Select several guard cells to draw. Include the stomate and the 2 guard cells in each of the drawings. Remember that also the stomate is bordered by a pair of specialized guard cells that you can see here, and they regulate the site of the stomatal opening. Once again, the air enters the plant through the stomatal pore and contains carbon dioxide, which is used in photosynthesis. And to have photosynthesis, the guard cells need to have chlorophyll. And the oxygen, which comes from photosynthesis, is used for respiration and is produced as a product of this process. Also, water vapor exits the leaf through the stomatas, and this is called transpiration. We need to report our findings now. There is a format we will use. The format is as follows. Use an introduction with a purpose. List materials. Follow this with a procedure so others can replicate the experiment. Then record the results, which include drawings and structures. List their functions. Lastly, write a conclusion, generally addressing your findings, and include if you proved the purpose. You need to know if you understood the functions of the guard cells. Just restate the purpose. Wow, another awesome experiment. Now it's time to put away our microscope. First, we turn off the light. Then we take off the glass slide from the stage, followed by putting the ocular lens in 4x facing you, and then removing any spillage of water from the stage. Clean it off well. Clean off the ocular lens. You might use a soft towel and lens paper. all ready to cover it up. Wow, this was really awesome. See you later.